Hello, everyone. My name is Vida Jamali. I'm a postdoctoral researcher at University of California, Berkeley, and today I'm going to talk about using deep neural networks for understanding anomalous surface diffusion of nanoparticles in liquid cell transmission electron microscopy. Visualizing nanoscale dynamics and structure in complex material systems is important for us because not only would it enable us to understand the underlying physics of these systems through connecting nanoscopic phenomena to micro and macro scale properties, but it would also allow us to engineer new materials, new synthetic chemical materials. There has been a lot of efforts in developing microscopy and imaging tools. For example, optical microscopy has enabled us to process new materials, understand biology, but it is limited with the optical diffraction limit or Abbe diffraction limit, which basically says that we cannot resolve features smaller than 250 nanometer using visible wavelength light. So now there has been advancements in pushing down this spatial resolution down to 20 nanometer, for example, with super resolution microscopy techniques, which was awarded the Nobel Prize in Chemistry in 2014. But the issue still that remains is that the temporal resolution is not that good, beside the fact that we cannot resolve features in dynamics such as conformational changes or rotational dynamics. So liquid cell transmission electron microscopy is a technique that allows to observe nanoscale phenomena with a spatial resolution of a transmission electron microscope, but also with a temporal resolution of millisecond. And this technique has been developed and has advanced in the last 15 years. When basically what it does is that we have this microliter of a liquid containing our nanoscale objects and entities that are now encapsulated in this nanofluidic chamber that has transparent windows made of silicon nitride or graphene, which is transparent to the electron beam. And as we shine the electron beam through this chamber, then we can observe the dynamics that is happening inside this chamber. So now, in order, this technique has been developed and has been used as an observational tool for studying nanoparticles growth or interactions between nanoparticles. But if we want to use this technique for looking at the dynamics, for example, diffusive motion of nanoparticles in boundary layers, or for techniques such as nanorheology to look at the motion of these particles in fluid environment with different rheological properties, there are still questions that we need to answer before moving forward with this technique. And that is, how does the electron beam from a microscope modifies the anomalous or the motion of general motion of these nanoparticles in a liquid cell environment close to the surface? And are they moving in a Brownian way or in a non-Brownian anomalous way? And what does this type of diffusive motion that they manifest tells us about the material properties of the local environment? To answer these questions, we have some challenges, and one of them is the limited number of nanoparticles. So if you are trying to use this technique for single particle tracking, so we want to keep avoid any interparticle interaction, so we want to keep the total number of particles low, so we have a dilute solution. And that means we don't have access to a large number of nanoparticles that we can collect data from. Also, there is data size limitation. So we have the state-of-the-art cameras are right now producing data with a rate of 3 gigabytes per second. And that means that, let's say we are collecting data from 20 particles in an in situ experiment that are each 1 minute to 2 minutes long, and they're collected at 100 frames per second. And that is equivalent to 2 terabytes of data. So we have these two limitations, which both means that we do not have access to a large ensemble of nanoparticles, and so we cannot use ensemble averaging statistical methods for analyzing the data. What we are trying to do here is that we're trying to answer these questions considering these challenges by looking at a model system of gold nanorods that are 60 nanometer long, and they're dispersed in water, and as they diffuse near the silicon nitride membrane of a commercial liquid, so we're trying to analyze their diffusive motion and understand what does the electron beam do to the system. So let's take a look at the two videos from our example videos from our in situ liquid cell TM data. One is at dose rate of 15 on the left-hand side, and the other one is at dose rate of 35 on the right-hand side. We can see on left-hand side, the particle is barely moving. It is moving, but it is very limited. But on the right-hand side, the dose rate is increased. The diffusivity increases by a lot. And so now if we process these videos and track the centroid of this particle in time and plot them in the same time interval of 70 seconds to the scale to the same size, we can see that diffusivity increases as we increase the electron beam dose rate. But now if we magnify these two trajectories, we can see that it is not just the diffusivity that increases, but also the diffusion pattern changes as we increase the electron beam dose rate of the microscope. And so now that we know that there is something going on, the question we are trying to answer is, how does the electron beam rate, dose rate, affects the nanoparticles diffusion near the surface in liquid cell TEM? And this is a foundational question that we need to answer before moving forward in using this technique and the motion of particles in liquid cell TEM for understanding rheological properties of more sophisticated soft matter systems, for example, for something like nanorheology. So let's take a look at three classes of diffusion that are relevant to us. The first class is Brownian motion, where we know that it's caused by the thermal noise of the water molecules in solution. So the particle takes these completely random, non-correlated steps, and we plot the distribution of displacements for this particle. It is a Gaussian distribution. Now, the second class is fractional Brownian motion, specifically subdiffusive FBM, and this typically happens when we have a diffusion of a nanoparticle in a viscoelastic gel-like network. And this means that basically the steps that the particle takes are anti-correlated. So if the first step is in one direction, the next step will be anti-correlated with that, so that the particle doesn't diffuse overall a lot, and it's a subdiffusive motion. Now, the third class is continuous time random walk or subdiffusive CTRW, where this case typically happens when we have a diffusion of nanoparticles over an energy landscape with trapping sites, which are basically these potential wells with unequal binding affinities. And we are showing these here with the stars, and so the particle gets trapped in these stars, and every time that it gets trapped, it draws a waiting time from a heavy tail distribution of waiting times to decide how long it would stay on the current point before it makes the jump to the next point. And it also takes the step size from draws the step size from a heavy tail distribution to decide how big of a jump it should make in each step. And overall, these steps are all non-correlated. And what gives rise to subdiffusivity is just this waiting time distribution that is heavy tail. So now if we are given these physical parameters of the system, for example, diffusion constant, and given these physical models, we are asked to simulate a trajectory from each of these classes of physical models. The forward problem is pretty straightforward. However, if we are given an experimental trajectory, for example, from liquid cell TEM data, and we are asked to solve the inverse problem of finding the type of diffusion that we see in this experimental trajectory, is a very challenging task. And this because it's specifically challenging when you have limited number of trajectories, when trajectories are short, or we have hybrid behavior, presence of multiple behaviors in the trajectory. And this is also the case for liquid cell TEM, that we know that we don't have access to a large ensemble of nanoparticles. So what we are trying to do here is that use artificial intelligence tools, specifically deep neural networks, to solve this problem of classification, as well as determining how short of a trajectory we can collect from liquid cell TEM to still would be able to classify the type of diffusion that the particles show in a liquid cell TEM data. So this is an overview of our platform. We do this liquid cell TEM experiments where we are collecting tens of thousands of frames by like images. This is a time series of images that shows the particle's motion through time, and we process these images by finding, in this case, the centroid of the particle. We are interested in finding the translational motion of this particle. So we get to this experimental trajectory, which we can form this matrix that contains X and Y coordinates of the particle in time. This is the input for our MotionNet, which is our convolutional neural network, and we call it MoNet. And MoNet is trained on 10,000 simulated trajectories from three classes of diffusion, FBM, Brownian, and CTRW that we talked about. And the trajectories are all 300 frames long. And what MoNet does is that it tries to learn the filters and the weights associated with these filters, that finds the pattern in this training data associated with each of these classes of diffusion. The output of MoNet is the probability class, probability of the diffusion class associated with the given input. So that with what probability we have continuous time random walk, FBM, or Brownian motion for each trajectory that we are testing this on. So now, if we look at what MoNet is doing in learning from the data, it takes basically this trajectory vector, it makes this trajectory increment vector dx, and it tries to find the convolution of this trajectory increment vector with these filters with size, for example, k equal 3, and the dilation of 1, which shows how many steps it's looking at the same time. And it can do that for multiple dilations of, for example, 1, 2, and 4, which are we have chosen 2 to the power of n, inspired by a canonical statistical test, to find multi-resolution, like basically long-term phenomena that happens at different resolutions along the trajectory. So if there's any long-term correlation in the data present. And we do this with different types of filters. And this is only the first sub-layer of our first layer, but we have 6 of these layers that are working in parallel, and each of them have different filters with different dilations to capture different type of patterns along the trajectory. And the result of all of these get concatenated into this vector that is then fully connected to our subsequent layers. Our final layer is we apply a softmax function, where it's basically a normalized exponential function that would give as an output a probability associated with each of these diffusion classes. We have tested the performance of MoNet for different trajectory length on 10,000 simulated scene set of trajectories for each of these trajectory lengths. And what we can see is the validation accuracy at trajectory length of 300, which is what we have used for data, is about 90%, and it doesn't improve that much by increasing the trajectory length. So this would then tell us like this 300 is short enough to be still able to distinguish what type of diffusion we have in our data from liquid cell TEM experiments. Now, if we apply this model on our data that is collected from dose ranges of 2 to 49 electron per angstrom square per second for 373 mini trajectories that are each 300 frame long from 30 nanoparticles. And these pie charts are showing the average behavior for each particle. And we can see that there is this crossover in the behavior from fractional Brownian motion to continuous time random walk as we increase the electron beam dose rate in the TEM microscope. And so now that we know what is the prediction for MoNet, we can also benchmark these results of MoNet with the canonical statistical test. The first test is looking at the distribution of displacement. And as we talked about it earlier, for low dose rate where we have FBM, we would expect that this should fit well with a Gaussian curve. And this is indeed correct. And for higher doses where we have the CTRW behavior, we see a power law, heavy tail power law distribution for the displacement distribution. Now, if you look at the time average mean square displacement versus time delay for our data, at low dose rates, we have a sublinear slope, which indicates that we have FBM present in our data, FBM behavior present, but at higher dose rates, the slope is 1, which in this case means that it is a non-ergodic continuous time random walk process because this is time average MST curve. And so now the last test is the p-variation or total variation test, which is a canonical test that was developed in the 1970s. And this would basically tell us if we have anti-correlation present in data, which we can see that it is present at low dose rates where our prediction is for FBM behavior. And at a higher dose rate, this becomes independent or non-correlated, which is what you would expect for a continuous time random walk behavior. So now that we know that what sort of behavior diffusion pattern we have in the system, the question we are trying to answer is what gives rise to these diffusive patterns in terms of material properties from a chemistry perspective, because at the end of the day, we are interested in developing microscopic models that would explain this diffusion in this system. So let's go back to the definition of FBM, where we would expect that the particles show this behavior when we have a viscoelastic environment. And so what we think is happening, the hypothesis is that we have this silicon nitride surface, which is decorated with silanol molecular species. And so these form these potential wells that have unequal binding affinities. At low dose rates, the particle gets trapped in these potential wells, but the thermal energy is not large enough to take it out of these potential wells. So it experiences the local material properties of the environment inside one single potential well, or maybe a few neighboring potential wells. And in that case, within few nanometers of the surface, we have these hydrogen bonded water molecule layers, which gives rise to viscoelasticity that it would experience within this potential well. So that would explain our FBM behavior. But now as we increase the electron beam dose rate, there's the radiolysis happening in water, there's the pH changes, and we have these radiolysis species that are passivating partially these silanol molecular groups on the surface. And so they're smoothing out these potential wells. Now the thermal energy of the particle is large enough to take it out of these potential wells, so the particle can experience multiple potential wells over the silicon nitride surface membrane. And so now we have the presence of CTRW behavior. So even though these two scenarios are explaining the crossover in the behavior that we observe, we want to note that there is also the possibility that we have the coexistence and basically superposition of both phenomena, but at different time scales. And we are currently working on developing theoretical Langevin-based models that would explain these multi-resolution phenomena. And with that, I want to thank you all for listening. And I want to thank Oliver Stoddard's lab, and postdoc lab, and Kranti Mondadopu's lab at UC Berkeley and the NSF for funding this project. I would be happy to answer your questions. Thank you all.